Hi all, I hope everyone is having a good health. My name is Omer and I'm working as a PhD candidate in the Department of Pathology, Norwegian Radium Hospital, Oslo, Norway, under the supervision of Dr. Jumbai Wang, who is the principal investigator of this project and of the group as a whole as well. I'll be presenting my software project, a package, clustering of position-specific wet matrices within the various protein families. With the advancement in information technology, new high-throughput methods have produced different collections of transcription factor binding motifs, which has produced redundant entries in databases that has been generated from various resources. So there is a need of automatic way of clustering or making groups of similar looking transcription factors so that they can be used as a group by finding their consensus or a representative motif. So here we present a software package for clustering the transcription factors. And after clustering, we estimate and calculate a consensus PWM for every cluster as well. Initially, we assign a DNA binding domain family label to every input PWM file, and then we classify them in their respective DBDs, and we apply the clustering within each DBD. After clustering, we also have the quality cluster assessment through different statistical methods. Here is an overview of the methods and the materials that we have used within our project. So initially we take position wet matrices as input files, and we classify them with the different DNA binding domain family name. Before classifying them, we also have produced a uniform database having the same name for every DPT, because different sources are using different nomenclature. So what we have used different sources, and we have given them a uniform name and used that uniform named database for our classification. After classifying them in their respective DBD folder, we apply clustering to each of the DNA binding domain family and make clusters. During the clustering process, when the clusters are made, we also calculate the consensus motif of every cluster. The output of clustering is then subjected for the quality assessment, and we can also plot the clusters as well by looking into the motifs and the representative that has been calculated for a cluster. Here is the overall flow of our project. We also get reports of every step in text file. So before classifying, we make the clean database, a uniform database, and export it to a text file. Then we have summary of classification, clustering, quality assessment in the text files for user references. There is a module in the package that helps us visualize the clustering results. So this is how a cluster looks like. It gives us the information about the DBD, the DNA binding domain, which is in this example HMG, and the cluster number 6. So it plots all the motifs along with its representative on the top. Quality assessment of the clusters made is not as straightforward, and it cannot be assessed with the single statistical factor. So we introduced a unique method of quality assessment and finding out the unknown or wrongly clustered PWMs. As here this slide shows the results of HMG DNA binding domain family of every cluster and its its mean values, and the range of similarities it have. Similar results can be produced for every family. The software package gets input from the command line from the user, and it produces the results accordingly. For questions and collaborations, please reach us out at the email address given in the slide. You can also have a look at our work in the link given. We are thankful to the Southeastern Norway Regional Health Authority and the Norwegian Research Council. That's all from my side. Thank you for listening.